Today's presentation is about chromosomes. These are physical objects on which genes are placed. We are going to explain what are chromosomes, what's the relationship with DNA as a type of chemical compound, and what we can learn from them. If you want to know more, please download the presentation from the disk. Then you will find the link. There is the link for the movie you can see in the web. However, I cannot include it in this presentation because I cannot record it together from two sources. So let it be. First, we have to recall what is a gene. A gene, as you should remember already, is a particular sequence of DNA encoding for some protein. So a gene has a physical form of nucleotides. One by one, they encode for the sequence of amino acids in the protein. And the genome, which is also a term we are going to use, is the whole DNA of the organism. Most of this DNA will be enclosed within the nucleus. These two terms must be understood. So once more, a gene is a sequence of DNA encoding for a protein, and genome is whole DNA of the organism or the cell. You already know that DNA has a specific physical form. It is a polymer made of many nucleotides, placed one by one. Looking at bases in these nucleotides, you can read the sequence. Physically, most of DNA is within the nucleus, of course in cells which do possess a nucleus. In prokaryotic cells, DNA will float just in cytoplasm, but now let's focus on eukaryotic cells. They got a nucleus, and inside there is most of cells' DNA. The rest will be in chloroplasts and mitochondria, but we are not going to discuss it. So for our use, we can assume that the whole DNA is in the nucleus. And due to the fact that this is a particular chemical substance, you can look at the single molecule of DNA. Such a molecule will have a shape of double strand. And if you look at DNA which is in the nucleus, a physical piece of DNA, such a double strand which can be distinguished from other pieces of DNA, is called a chromosome. Chromosome is a molecule of DNA. And what we have to know is that in the nucleus of a single eukaryotic cell, there are always many chromosomes. What means that the whole DNA which is enclosed there is not in the form of one double strand, but many pieces. A chromosome is not pure DNA. The molecule of DNA is the most important because it encodes proteins, but it must be somehow physically arranged. So, as you can see on the diagram, the double helix of DNA is not allowed to float freely, but it is organized in specific structures using proteins called histones. I'm not sure if this name is crucial. A histone protein is something what you not necessarily have to memorize, but what you have to memorize that together both of these proteins and DNA wrapped around them will form a structure which is called a nucleosome. So actually, if you look at DNA floating in the cell's nucleus, it will have a form resembling the string of beds. Nucleosomes one by one, joined by the short fragments of loose DNA which is not wrapped around any protein. To sum up, DNA in the nucleus is arranged in special structures called nucleosomes, and this arrangement gives the DNA the form resembling the string of beds. A long time ago, when people were not aware what actually is present in the cell's nucleus, they just could try to guess looking at pictures which were present under the microscope. What they could see was a sort of messy substance without any specific structure, but if treated with specific dyes, it could get some color. From the Greek chromos, this is what gives the name to this substance which is created by associating DNA with proteins. It is called chromatin. So, the term chromatin is used to name the whole DNA in the nucleus together with protein it is associated with. So, this is a DNA double strand. These are nucleosomes, and altogether, the whole string arranged into nucleosomes, DNA with proteins, is usually called chromatin. I'm not particularly fond of this term, but sometimes it is useful. For instance, if you want to describe the fact that in a cell, when you look at the nucleus, it resembles just a shapeless mass. Chromatin is a term which you can use for the whole DNA together visible as this grayish mass in the nucleus. However, this is not what you can always see when you look at the nucleus. Because this form we described, DNA arranged just into nucleosomes, and then these strings of beds floating in the nucleus, it happens only if cell is not dividing at the moment. It is called resting cell, or sometimes a non-dividing cell. In this stage, this is exactly what you would find looking at DNA. But when a cell is going to divide, it will prepare for this process by performing something what is called supercoiling or chromatin condensation. Supercoiling, as you can see, is taking these strings of beds made of DNA and proteins and twisting it into something what has also its own name, but you don't have to memorize it. And then this structure is twisted once more and once more, finally producing such a structure. So from extremely long and thin string, finally we've got rather short and fat sausage-like structure produced, which is much easier to manipulate. And this is actually the point of this whole supercoiling. When the cell is going to divide, the crucial focus of the cell is to segregate all DNA molecules properly to the resulting cell. It would be very hard if you try to do that manipulating with long and thin tangling strings. So condensation of chromosomes to such structures helps to manipulate with them. There is also a drawback. At this stage, chromosome cannot be read. DNA is packed in this stage, locked, sort of, so it cannot be used in any other way than just transporting. So we can assume that actually DNA may be present in the cell in two forms. If a cell is not dividing, DNA will be in the form of loose chromatin and can be read. While when going to divide, DNA is folded into so-called division chromosomes, and in this stage it cannot be read but can be transported easily. And you cannot see any structures in the loose chromatin in the cell looking under the microscope. DNA strands are too thin to be seen even under the microscope. But if they condense to this shape, then they can be spotted easily, especially if dyed with a special pigment. The consequence is that for a long time it was thought that these chromosomes are just absent in non-dividing cell, and then they appear when a cell is going to divide. No, they are present in the cell all the time. They just become visible because they change the form into which DNA is organized. But the chromosome is just a molecule of DNA. So if packed or just loose, it is still a chromosome. If you can see it or not, it doesn't matter. You are also expected to know some terms describing the structure of such a chromosome. However, not all these names which are visible here are obligatory. So try to focus and you will learn which names should be known to help you to describe what happens to such a chromosome. Each chromosome you can see during cell division is usually in the shape of such a short fat X. This shape results from the fact that DNA is replicated shortly before the division, what actually means that one half of a chromosome is identical to another half of a chromosome because they are the identical copies. So what was produced by DNA replication is now held together by the structure which is called, and this is the name you have to remember, centromere. A centromere is the place in which two identical copies of DNA are held together after DNA replication, not to get lost. So this is like when DNA is copied, these two copies should stay together. They are clipped by a centromere, and they will split only at the proper moment to make sure that DNA is correctly segregated to resulting cells. Such a one half of a chromosome is called chromatid. This is one chromatid, and this is another one. So half of a chromosome is chromatid, and the place they are connected together is called centromere. It may be also quite useful to know that these parts which stick out from a centromere are called arms. This is an arm, and this is an arm, and this is an arm, and this one is also an arm, but you don't have to know their names like P or Q. It is not important, and I truly doubt if anybody will ask you about more details in the structure of chromosome than that. So once more to repeat. Centromere, holding two copies of DNA together. One copy is one chromatid, one half of a chromosome. And at the opposite side, another identical copy in the form of the second chromatid. Two chromatids clipped with a centromere form the whole division chromosome in the shape of X. In a nucleus of any eukaryotic cell, there should be a correct number of chromosomes. This is so-called typical species feature. It means that each species will have a typical number of chromosomes that should be found in the nucleus, and a different number means some abnormality and usually leads to some health problems. As I said at the beginning, in such a nucleus, there is never one molecule of DNA, not one chromosome. There is usually some particular number of them, and what you also can see on this diagram is that actually they can be grouped in pairs. What you see here is called a karyogram, the picture of a specific idea. The whole set that should be present in the nucleus of the eukaryotic cell, the whole set of chromosomes is called a karyotype. So this is sort of the idea. I can ask you a question, what is a typical karyotype of a human? And then you should be able to say, typically there are 46 chromosomes, because this is what a human karyotype is like. 46 chromosomes arranged in 23 pairs. But the picture made of that is called a karyogram. This is a physical picture, and of course it must come from a particular named cell of a particular individual. So this is no longer an idea. This is the real photo of real chromosomes. How is it made? You have to wait for a proper moment during cell division. You make a photo of a proper, well-visible nucleus, and then you physically cut fragments of the photo containing a picture of a chromosome, and you try to arrange them in pairs. This is more or less how it is done. If you look at such a human karyotype, you will see that these chromosomes are arranged in pairs. This is not accidental. It comes from the fact that if an organism is reproducing sexually, there is a typo, I'm sorry. If the organism is reproducing sexually, one set of chromosomes will come from each parent. So actually, we are not having one set of chromosomes. We are having two of them, each of them obtained from one of parents. One of your parents gives you one complete set of 23 chromosomes. And another parent gives you the set of similar 23 chromosomes. So if you get 46 of them in each of your cells, it actually means that this is not a single set. There are two sets due to the fact that we've got two parents. And chromosomes similar enough to form one pair, equivalent chromosomes from two different people present now in your cells, are called homologous chromosomes. Can you see what makes them similar? Their length, this is one criterion, and the position of a centromere. Some of the centromeres will be in the middle, some of them much closer to one end. Due to that, and sometimes also due to the pattern you can see on them in specific staining, you can check which chromosomes belong to the same homologous pair. What does it mean that they are homologous? Actually and physically, it means that they contain the same set of genes. On the homologous pair of chromosomes, you will find exactly the same set of genes at the same places. So let's say that here you can have the gene for eye color coming from one of your parents. And the second chromosome will have exactly the same gene at the same place. If you've got a gene for amylase, the special enzyme digesting starch, it would be, let's say, here on one of these chromosomes, and in a homologous one, the same gene should be exactly at the same place. So this is how sexual reproduction works. Your parents have some chromosomes, and when reproducing, they pass one of their sets to the offspring. Another parent provides another set. This set should contain all necessary chromosomes, and it leads to the fact that the complete set contains double number of them. Equivalents containing the same set of genes will form a homologous pair. So when you look at the whole karyotype, you can expect that there is a specific set of genes placed on the first chromosome. This and this chromosome will contain the same set of genes, however, not necessarily identical in their structure. They can be mutated. But the same type of gene will be always present at the same place. But when you look at the second chromosome, it will contain different set of genes than the first one and different than the rest of them. Each of chromosomes will have its specific genetic content different than for other chromosomes. The number of similar homologous chromosomes in the set finally results in the idea of ploidy. Something can be haploid. It means that there is only one chromosome of each type, or something can be diploid, what means that each chromosome will have one homologous equivalent, so finally they will form pairs, and the whole set will be doubled. And sometimes it can be triploid, quadruploid, however, these are rather rare cases. If it comes to haploid and diploid cells, in sexually reproducing organisms, there is usually relatively simple rule, however, not in all species, but you can generalize it because this is true for humans and many other creatures. It is like that. All cells of our body, so-called somatic cells, are diploid. They will contain double set of chromosomes, each of them coming from another parent. But there will be also a specific type of cells which are produced only to reproduce. They are called gametes, sperm cells and egg cells. Due to the fact that they are going to be used for reproduction, they have to contain only haploid set of chromosomes, because their aim is to be combined with another haploid set coming from another parent. So if the organism is going to reproduce sexually, a specific cells will be produced called gametes, each of them containing haploid number of chromosomes. And actually for most organisms this is a simple rule. The whole body made of diploid cells. And only for reproduction the set of haploid cells is produced to enable combination with another haploid set. You are obliged not only to know the general idea of a karyotype, but also you have to know how the typical human karyotype looks like. So what should be known? You have to know that the typical number in the haploid set of human chromosomes is 23. And you have to know that in each somatic cell, diploid set is found, what means that finally each regular somatic human cell should contain 46 chromosomes arranged in 23 homologous pairs. They have specific length, specific structure, and if arranged in such a set called karyotype, you can analyze the set of chromosomes and check if everything is correct as it should be. Abnormalities, as I mentioned, may lead to severe health problems. When I said that a typical set of human chromosomes contains 23 homologous pairs, it was not fully true. 22 are always homologous. What means that if you look at chromosomes from 1 to 22, they always, in normal circumstances, will form homologous pairs. But this is not the same for the last pair, which is called sex chromosome. In this case, sex coding is performed by containing proper set of chromosomes for each sex. If the last pair contains 2 homologous chromosomes, so if they get a similar length and pattern of bands, it means that we've got a female. Such a long chromosome in the last pair is called X. And all females should possess 2 X chromosomes, so actually this is a typical homologous pair. However, in males, only 1 chromosome is of this X type, a long one. The second one should be much shorter and it is called Y. When you get such a set of chromosomes in your cells, then you develop into a male. So this is also something you have to know. 2 X chromosomes and the organism develops into female. A human organism, of course, because in some other species it may be different. If a human organism possesses X and Y, it typically develops into a male individual. It should be like that. So looking at the karyotype of a person, you can deduce the sex of a person. It may be a task you are given. If shown such a karyotype, you should be able to actually see 2 aspects. What is the sex of such a person? And if the set of chromosomes is typical for humans, or maybe it contains some abnormalities. What could be these common abnormalities in chromosome number will be discussed separately, and later you are going to learn what types of chromosomal aberrations are common and what are their consequences. Right now you just have to know how the regular karyotype looks like. It is more or less everything what you have to know basically of chromosomes as physical structures present in human cells. Not everything what regards chromosomes was said today, because some aspects can be understood only if they are placed in proper context. So we are going to mention some important information about chromosomes later, talking about cell division, and more later talking about chromosomal aberration as a health issue. But for today, that's all. Thank you.